Most migrating cells place their centrosome in front of the nucleus, facing the direction in which the cell is moving. It's not clear why cells do this. Centrosome positioning may promote persistent directional migration by polarizing microtubules, which, in turn, helps organize other cytoskeletal networks and orient membrane trafficking toward the cell's leading edge. Sandrine Etienne-Manneville from the Pasteur Institute in Paris, France, has previously uncovered components of a signaling cascade that reorients the centrosome in astrocytes migrating toward a wound.
This involves activation of integrins at the leading edge of the cells, which controls CDC42, PAR6 atypical PKC complex. And this complex then allows the recruitment of two major proteins, which are APC and DLG1, at the front edge of the cell. So the question we had at that point was how this signaling cascade at the leading edge of the cells could actually modulate the position of the centrosome and how the microtubules, which are likely to play a key role in this, could be regulated by the signaling cascade.
The researchers began by looking at the organization of microtubules at the leading edge of migrating astrocytes.
There are several mechanisms that have been proposed to control centrosome positioning. One involves the anchoring of the microtubules at the cell cortex. So for this we use the turf microscopy because it allows to visualize a thin region of the cells in which microtubules go close to the basal plasma membrane. And this allowed us to really visualize the anchoring of the microtubules at the leading edge.
Microtubules reach down to the basal plasma membrane near the cell front, just behind the lamellipodia and an actin-rich adhesive zone. The microtubules bent as they approach the bottom of the cell and then curled back up at their plus ends.
Microtubules are very stiff polymers. The fact that they are bent like this indicates that there is a force exerted on the microtubules to hold them to the cell cortex. And that's why we decided to study dynein, because we thought a microtubule-associated motor would be able to mediate such a force.
The -N-directed motor dynein is required for centrosome positioning in a variety of cell types. The motor could anchor microtubules at the leading edge and haul the centrosome into position in front of the nucleus. Turf microscopy revealed that dynein was present on microtubules at the cell front. As well as misorienting the centrosome, dynein depletion blocked microtubule anchoring and dramatically disrupted the cytoskeletal network. In control cells, microtubules grew directly toward the leading edge and depolymerized when they reached it. In the absence of dynein, microtubules grew in all directions, turning as they met the leading edge to produce a disorganized network of buckled filaments. So dynein-mediated anchorage is required to organize leading-edge microtubules and position the centrosome. But what lies upstream of the microtubule motor?
When we knock down molecules of the CDC42 signaling pathway, CDC42 or DLG1 knockdown induces a complete disruption of the microtubule network, which is almost similar at the one we observe with dynein depletion. The microtubule network is completely disorganized, and there's no more polarized anchoring.
Leading-edge microtubules weren't anchored because they didn't recruit dynein in the absence of CDC42 or DLG1. The researchers then turned their attention to GCAP, a protein that combined both DLG1 and the dynein light chain subunit LC8.
We looked whether GCAP was involved in the interaction between dynein and DLG1, and we found that if we deplete GCAP, there is a strong inhibition of the interaction between DLG1 and dynein, indicating that GCAP is probably a major mediator of DLG1-dynein interaction.
GCAP localized to leading-edge microtubules upon wounding, but it wasn't recruited to the cell front if DLG1 or CDC42 were depleted. GCAP, in turn, was required for dynein recruitment and microtubule anchoring, so that, in the absence of GCAP, microtubules were disorganized and centrosomes weren't oriented in the right direction.
So what we think is that following wounding, there is an integrin signaling pathway that leads to CDC42 activation and recruitment at the wound edge of the cells. This leads to the activation of the PAR6 atypical PKC complex, which controls the recruitment of DLG1 to the cell front. DLG1 has two functions. First, it interacts with APC, and that may mediate an initial capture of the microtubule placents. And on the other hand, DLG1 interacts with GCAP, and this leads to the recruitment of dynein on these leading-edge microtubules. dynein then is responsible for the anchoring of the microtubules and probably promotes the force which allows the reorientation of the centrosome in front of the nucleus. We think that such a signaling pathway could be involved in microtubule reorganization and centrosome positioning in other organisms and cell types, not only in cell migration but also in cell division and other processes.
For dynein to anchor microtubules and generate a pulling force on the centrosome, it must somehow associate with the leading-edge cell cortex.
So this is something we would like to investigate. We don't exactly know how dynein interacts with the cellular cortex. In addition to this, there's of course the role of centrosome positioning, which is still not clear during cell migration, so that would be something very important to carefully study. And the other thing we were very interested in looking at is how alteration of the signaling cascade could be involved in the abnormal polarization of cancer cells. We would like to study glioma cell migration and look whether in these cells in which centrosome orientation doesn't occur normally, this signaling cascade is altered.
In the meantime, you can read more about the pathway that recruits dynein to leading-edge microtubules to regulate centrosome positioning during cell migration in the paper by Manneville et al., published in the November 1st edition of the Journal of Cell Biology.